Thank you all very much for coming today. I'm really happy to introduce Caleb Kemere as our speaker today. Quick introduction about Caleb before he gets started. He did his MS and PhD at Stanford in the EE department, working with Teresa Ming. He started out doing mainly DSP for wireless applications, ended up moving over more towards algorithm development for neuroscience and neuroprosthetics. For the past 4 years, he's been a postdoc at UC San Francisco, studying the brain with a couple different tools, including neural recording and also a new technique, optogenetics, which I'm really excited about hearing about. So, Caleb.
Well, thanks, guys, for having me. There'll be a little bit of sort of talk about the brain at a high level today, which we agreed was appropriate for April 20th. And that will be my only reference to today's date. So why should we, so I'm going to assume that most of you are engineers, and if there are neuroscientists in the audience, I apologize ahead of time for the lack of detail and the huge jumps that I'm going to make. But hopefully there aren't very many of you who will correct me, but feel free to if you feel it's necessary. So why should we as engineers be interested in studying the brain? I think there's two main reasons. The first may be obvious to many of you, that there are things that we do with our brains that we would like to learn how to do with computers, but we don't know how to. So for example, speech processing or detecting facial expressions or gestures. And then at a level above that, when we have detected something, to be able to understand what it means. And then at a level above that, we'd like to be able to integrate that meaning into some context, some broader history of our own experience or our experience with somebody else. And sort of to top it all off, our brains can do these very complicated things while being powered with donuts. And so we have a lot to learn about low power. The other reason that we'd be interested in studying the brain as engineers is that we would like, in situations where there are malfunctions of the nervous system, to be able to repair them with technology. And there have been a couple of really successful examples of this, like cochlear implants or deep brain stimulators, and maybe on the forefront, artificial retinas. But the bottom line is that whether we're going to learn to mimic the brain or to repair it, the first thing we have to do is to understand how it works. Okay, so when do we know that we've understood some particular system in the nervous system? Well, I think that the principle that the science that I've been working on is that we know when we've understood something if we can interact with it, if we can prove causality at some level. So what do I mean by that? Well, at some level, this is our brain. It's just a really complicated input-output system with sensory input and motor output. So we can prove causality on the sensory side when we can take an artificial representation and encode it into neural signals that produce perception. So if we have, for example, a model of, say, visual motion that we think says something about how neurons in area MT in the cortex encode the perception of speed, then we can prove that that's true if we can inject signal into that area and produce the perception of speed. On the output side, we could imagine that we could prove the causality of, say, a belief about the motor cortex and the way that it relates to movement if we could decode neural activity in the motor cortex and use it to produce artificial movements. Okay, well, what about this stuff in the purple region, where we don't necessarily know what the representations are that we need to encode, and we don't, that we need to decode, and we don't necessarily know what the representations are that we want to write back in either? Well, it turns out that they're really interesting questions like cognition, maybe decision-making, and memory, which I'm interested in, live in here. And so in order to understand this region, we need to build theories, and then in order to interact and prove causality, we have to both decode the neural activity and then re-encode it in order to do this. And I think that these challenges are hard, and they require good engineering in order to build systems that will be able to interact with the brain at that level. Okay, so this is the sort of the organization of my talk. I'm going to be talking about a grand challenge, which is understanding memory today. And so for you guys, memory is stored as some distributed representation in your cortex. That's the newest part of your brain. It's the outside part. And it's generated there. It's sort of written to your cortex through a memory center that I'm going to give you a brief tutorial about a little bit later. So why is it interesting to study memory? So I'm going to give you those two categories again. Memory is interesting because it's able to be very rapidly formed. So presumably after my talk, you'll remember something from it. And yet you've really only heard me talk once. And so that's sort of remarkable that after one presentation of an event, you can remember it much later. There's this process of consolidation from short-term memory to long-term that can last for years. And then there are aspects of the recollection of memory, novelty detection of navigation that are all things where it may be that there are computer systems that we can improve by understanding how our brain performs these tasks. On the other side, malfunctions in this memory circuit presumably lead to deficits in learning, certainly memory loss, and also, interestingly, diseases like depression and general anxiety disorders also seem to be related to malfunctions in the circuit. Okay, so in order to build an interface that's going to interface with memory, we need three main blocks. And so this is sort of now the engineering organization of my talk. I'm going to talk about these three problems. The first problem is, how do we acquire the useful signals from the brain? What are they? And then how do we detect complicated hidden states and then decode information from that neural activity? And then what sort of tools could we use to re-encode that information into something that then will allow us to manipulate the memory system? Okay, so here's the outline. This is the order that we're going to go in. I'm sorry it's not clockwise or counterclockwise. It's just complicated. We're going to start by talking about what neural signals are and how we acquire them, and then the engineering challenges associated with that. So there's a lot of neurons in the brain. That probably is something that you already knew. And they communicate with each other using these electrical pulses called action potentials. Now, it turns out that because neurons are organized so that neurons which do similar things are in similar places in the brain, that if you observe the average activity of a large volume of tissue, you can sometimes decode useful information from that. And that basically is the principle that functional magnetic resonance imaging, external EEGs like you might get at your doctor, or these electrocorticograms that are basically large electrodes that go right inside your skull, this is the principle on which they're based. But the actual signal is being generated and being embedded in the activity of individual neurons. So for my talk, we're going to focus mainly on how to extract information from individual neurons. And the best way to do that is to use extracellular electrodes that are implanted chronically into the brain. And if you have many of these electrodes, then you can capture the information from many neurons at the same time. And that sort of maximizes the SNR of your ability to read out information from the brain. Okay, so I'm going to talk to you today about two different methods that we use for recording large number of neurons. And this is just a picture so that you can see it. The first system is a micro-machined electrode array. It's a 10 by 10 grid, sort of a bed of nails, and it's 4 millimeters on a side. So something about the size of your pinky fingernail. And we use this for extracting information from the cortical surface. And the example I'm going to give to you is from my graduate work, where we were recording information from the motor cortex of monkeys, motor cortex. Then the work that I've been doing more recently is in rat hippocampus. So this is a section of a rat's brain, and this dark stuff here is the cells of the hippocampus. They're very dense. And so what that means is that there are many cells that are very close to each other, and so if we want to differentiate their activity, instead of using single shanked electrodes like we do here, we use bundles of 4 wires. And because this is deep in the brain, we use what we call a micro drive. So each of the tetrodes is sort of, the depth is independently adjustable. But with both of these systems, we can record a few hundred neurons or several dozen up to 100 neurons simultaneously. So if we looked at a single one of these electrodes, what does the signal look like? Well, the first thing that we'd see is this sort of broad oscillation. And that's called the local field potential. And it turns out this is the same thing as you record with an EEG electrode on the surface of your skull. There are these voltage changes, so that's the word potential, voltage changes. But this is local to the area of the electrode. So at some level, this represents that summed activity of a lot of neurons, but now it's a lot of neurons local to the area that you're recording. And it turns out that there is some important information that is stored in the patterns of activity that we see in the local field potential, in addition to the activity of individual neurons, as I mentioned a second ago. So just hold this thought. So a good neural acquisition system is going to store this, but because it's such a low bandwidth, this is usually not a challenge for our recording systems. Okay, so if we cut out that local field potential, what we're left with is this noise or sort of indistinguishable neural activity, on top of which we can see the action potentials, the spikes of neurons. So the challenge then is that the signal that we want is the times at which individual neurons produce their action potentials or their spikes. So the signal that we want is the spike times. And this is the first engineering challenge that I'm going to talk to you about. So this is extracting the times that individual neurons are spiking from this activity. Now, what's complicated about this is that we have no way of aiming our electrodes at a particular neuron. So what we see is we see the activity of many neurons. So here, for example, are the waveforms that we might see if we sort of zoomed in and looked at each of these individual traces. There's a large waveform, and then there's a smaller one, and then there's some smaller ones, and then there's this stuff that's noisy. And we can detect these by just picking a threshold and saying, okay, well, we're going to look at the waveforms of things that cross the threshold. So the challenge of spike sorting is to pattern match the waveforms that we see, that we record in this activity, and identify them as as one neuron or another. So in this case, we've isolated by their shape 4 different neurons from this signal. Okay, so when we do this with this array, we can see maybe 1 to 2 neurons on average per electrode. So remember, we have 100 electrodes, so we can get upwards of maybe almost 200 neurons that we can record simultaneously. When we do this with tetrodes, now we have a signal on 4 channels, and so we can actually see maybe 2 to 10 neurons per tetrode, but because we have fewer tetrodes than we had electrodes, we still end up with something about 100.
Can I have a question?
Oh, yeah, of course.
Can you tell me the time scale on that spike profile? 25. 25 milliseconds, is that right?
No, sorry, that's incorrect. This whole thing is this whole thing is 1 millisecond. The scale is wrong. Yeah, sorry. This is 40 samples at 30 kilohertz, so a little bit more than a millisecond. This, however, is 0.1 millivolts. Sorry, these are very short events. So the local field potential was something that had an energy at about 400 hertz or lower, and now the spikes have energy in the kilohertz range, so maybe 3 kilohertz. Yes?
So you've shown 2 different spike trains there, the green and the red.
Yes.
And you mentioned that you sort based on, or you recognize based on sort of pattern.
Yes.
Is this because a neuron is going to have a habitual pattern of what its response is going to look like?
Sorry, yes. So when these electrodes are chronically implanted and they don't move in the tissue, then the shape of the action potential that you record on a given electrode doesn't change. That's not true. Actually, the process of gliosis and the degradation of the recording will cause you to lose signal over time. But on the timescale of a day or 2, the signal is stable. Yes?
So that you're recording your output of neurological activity.
Yes.
How are you conveying any kind of input so you have a controlled
Oh, you've got to wait for another 20 minutes. All right. So let me talk. So remember, this is our challenge number 1, our engineering challenge number 1 for neuroscience is spike sorting. So I consider spike sorting a 2.5-step process. So there's the initial training part. So given some sequence of data, figuring out what shaped action potential corresponds to the same neuron and which shapes correspond to different neurons. Then there's the simple ongoing process of, as you record, seeing an individual action potential waveform and assigning it to a particular neuron. And then for situations where the electrodes are moving, for example, and the shapes of these action potentials are changing over time, then the ideal spike sorting system would sort of track those changes and keep the proper identity for the neuron as its spike waveform changed slowly. So at Stanford, we set up a system that would do this initial training and this assignment process automatically. And the basic algorithm was noise whitening the background, dimensionality reduction like a PCA algorithm. And then for this initial training, there was a Gaussian mixture model for the clustering. OK, so this work, ideally for the kind of systems that I'm going to be talking about today, we want something that can work in an embedded context over a long period of time, hours, potentially days. And so we would like to take this system that we implemented in especially the part of spike sorting that we implemented in a small cluster of computers and implemented in the context of something that was small enough to be implantable, at least the part spike assignment and tracking. And the problem becomes more complicated when we think about tetrodes. So now here's tetrode recordings. So now remember that you can see a given neuron, and you can see its neural signature on 4 channels. And so the cool thing about tetrodes is it allows us to distinguish now the patterns of more neurons than we could have if we had just been recording with one electrode. If we had just been recording with this electrode, for example, then all 3 of these waveforms would have been thought to come from the same neuron. But because this red waveform because this orange waveform is small here and large here, we can distinguish them. Now, so obviously, clustering in this space is roughly 4 times harder than clustering in an individual neuron space. But what we're moving to in the lab are electrodes that have not 4 channels, but 8 or 16. And now, instead of knowing some set of electrodes that we might see signal on, it can be spread across many of them. And so the challenge of having an automated system that can do this in real time is one that I think will require some solving. I think it's an interesting question. And I think it would be a very useful tool to develop for neuroscientists. It's actually one of the sad things that when I, as an engineer, come to a neuroscience lab, I'm faced with a computer where I have to do this manually. And I think, oh, why can't I go to SourceForge and download a system that will do this automatically for me? Surely somebody has done this. But this sort of collaboration, unfortunately, has not spread in neuroscience. But I'd like to spread it. OK. So neural signal acquisition, summary. Here's the challenges. We would like to do spike detection. We need an architecture that we can implement in an implantable system. We would like to be able to do the detection of the clusters, the different spike waveforms, in a way that allows for iterative changes over time and for tracking and works for multiple electrodes. And I didn't actually mention this, but it turns out that sometimes neurons have particular associations between when they fire and different frequencies of oscillation in that local field potential that I talked about. And ideally, rather than having to, a week after you've gathered your data, run some large MATLAB script that extracts that, it would be really useful to be able to do this on the fly so that the important information can be extracted and then used in the experiment for feedback, as I'm going to talk about. So these are some papers that talk about the stuff that we did at Stanford on clustering and then on also sort of imagining architectures that would work in a silicon context for this problem. OK, so that was the acquisition of neural signals. So next, I want to give you an example of the kind of signal processing algorithm that we might use in the context of extracting information about hidden states from neural activity. And I'm going to come back to this point in the context of memory. But because I have a really nice complete story for my graduate work, that's what I'm going to present. So this is done with Teresa Ming and Krishna Shenoy at Stanford and with the people in their lab. I just want to thank them ahead of time. OK, so in Krishna's lab, the motivation was and still is a neuroprosthetic interface. Now, I realize that terminology can be confusing. So in the context of our research there, the goal of a neuroprosthetic interface is to help people for whom spinal cord injury or neurodegenerative disease have blocked the pathway of information from their intact brain to their limbs. So the challenge is to take information out of the brain, decode movement intent from that, and then use it to stimulate some sort of either natural muscle activity or perhaps an artificial limb movement, or in the simplest case, the movement of a computer cursor. So we focused in Krishna's lab on reaching movements. What do I mean by reaching movements? Well, I mean movements that are goal-oriented, like me picking up this glass. So it turns out that goal-directed movements, unlike sort of reflexive movements, take time to plan or prepare. And there's actually neural activity and neural processing that goes into generating the trajectories that underlie these movements. So in order to sort of accentuate that planning activity, we had our monkeys do a task which highlighted it. So here's the monkey that we were looking at their movements and their neural activity, and he's sitting in front of a touchscreen with his hand in the center. So a target comes up, and we train the monkey that he's going to move to that target, but he's not supposed to move until the target changes. It doesn't change color, it fills in. The target fills in, and then once it fills in, that's his movement, that's his cue to move, and so he moves his arm to the target. And then he gets a little drop of juice, and he moves his hand back to the center, and the task starts again. And we can get our monkeys to do 1,000 or maybe a couple of thousand of these movements a day, so we can get a lot of data. So what's cool about this task is that this period, where the monkey knows that where he's going to move, but hasn't moved yet, we assume that the neural activity that we're seeing corresponds to the preparation of his movement. And then during the period where he's moving, we assume that the neural activity that we're seeing corresponds to the generation of the movement. So what does the neural activity look like in these experiments? Well, so here's the firing of an individual neuron in an individual trial. The time scale is right this time. And here's the trajectory of his arm sort of in a radial direction. So the target comes on, and the neuron starts firing after some delay. Then a little bit later, here's the cue to move, and you can see the firing increases again, and then as he moves, there's this burst of activity. So we can see from this that there is some temporal dependence in the pattern of activity of this neuron. So there's nothing before the target came on, then there's some increase in activity, and then there's a second increase in activity in response to the go cue. Well, okay, so we couldn't really tell very much about the movement, just about the relationship of this neuron to the movement from that one trial. But fortunately, people and monkeys, when they're moving repeatedly to the same location, make stereotype movements. So the movement is the same, so we can assume that any variation that we see in the neural activity is coming from some sort of natural variation. So here you see neural activity over something like 50 trials to this target. And we can see here in the average of these, so the bend average probability of a spike, or the firing rate of the cell, that after the target comes on, there's an increase in activity. And then again, after the go cue with some latency, there's a second increase in activity. So we think of this as planning activity, and this is movement activity. And it turns out that this variation, the probability of seeing some particular example spike train for this particular movement, given this underlying firing rate that's time varying, can be modeled as a Poisson point process. Okay, so there's some time dependence of the neural activity that depends on the movement. There's some variation that's Poisson. What other variation do we see in the neural activity? Well, depending on which of these targets that the monkey is moving to, we see that the neural activity changes its amplitude quite substantially. So for example, here for movement to the right, we see that there's this large amount of planning activity. Here there's almost none. And then there's a very different sort of temporal pattern of movement activity for these two movements. So we might say that this neuron prefers rightward movements, for example. And we're recording from 100 neurons, or a few more than 100. And remember that this is a characteristic example, so this is how neurons fire, but they all have different parameters related to movement. Okay, so remember what we want to do is we want to decode the state of the neural activity and sort of perhaps some information from it. That's our goal. We're talking about the problem of decoding from neural activity. So here's a summary of the problem. For an individual movement, here is the pattern of activity that we see across 194 different neurons that we've recorded from our array. And here's sort of the time course of the movement. So today, the problem that I'm going to talk to you about is this. Given this signal, which we're going to model as a point process, a vector point process, so a vector across the dimension of the number of neurons that varies as a function of time, the challenge that I'm going to solve for you is how can we decode the target that the animal is going to move to, in this case, one of 8 possible choices, and the time that he wants to move. So there's sort of 2 classes of these sorts of systems in the context of a prosthetic interface. Some people are very interested in the exact time course of this movement. But for these goal-directed movements, I just told you that they were highly stereotyped. So that means that if you know where you're going to move, then you actually know the trajectory. So for these sorts of movements, all that we really care about is the goal and the time. Now, I should point out that, there is some variation, and this is sort of an interesting problem. And I'll talk to you at the end about the work that I've done related to that. OK, so the principle that we're going to use to decode is maximum likelihood.
I know you're more interested, or I think you said you're more interested in the action part of it, but in the planning part, have you noticed any difference between when the monkey first starts to learn this movement and when it becomes routine?
So actually, I'm very interested in, well, I'm very interested in planning activity. But this study was done on animals that were already really well trained. And so I don't really have access to that data. In fact, because the process of training an animal takes so long, we typically wait to do the implanting of the chronic electrode array until they've learned it well, so that we know that we'll have a reliable behavior. There's actually, I think, at least one animal that never could learn to do this, though it doesn't seem like that complicated of a task.
Okay. So for any rate, for your results, you're sort of in steady state.
In terms of learning, we're in steady state, exactly. But that reminds me that I did want to point something out. There are 2 regimes, there's actually 3 regimes of neural activity that we see here. There is some sort of baseline state when the animal's not moving. There's some sort of state where he's planning, and then there's some sort of state where he's actually moving. And if we were to just use a single model to decode this, then we would find that we had confusion during whichever period we weren't modeling. I don't know if that makes sense. Hopefully it'll make sense in a second. OK, so this was the state of the art when we started working on this problem. People would look at only this window of activity following the target onset in order to decode which target the monkey was going to move to. So one of the very first things that we said was, OK, well, there are definitely 2 different patterns of activity that correspond to movement here. So let's use a window of activity following target onset and a window of activity corresponding to movement to decode this. And so then you can imagine a very simple decoding algorithm. We're going to define a model where we say, let's learn the average firing rate over these windows, so the average number of spikes observed during the plan period, during the movement period for each neuron, so these vectors. And so we use some training activity to decide to find that out. And then on a given trial where we're trying to decode activity, we'll just say the target that we decode is the one that maximizes this a posteriori probability. So for that trial, the number of spikes in this period and the number of spikes in this period given the model. OK, so that obviously presents a problem. In using one of these prosthetic interfaces as a user, how would you tell the system when the planning period was and when the movement period was? So you could actually imagine something where you could do this. You could have like a little signal that went into the person's ear that said, OK, get ready, get ready. OK, plan a movement. OK, get ready, get ready. OK, move. OK, get ready, plan a movement. OK, but you can see that that's not what you want. You want something that automatically figures out which of these latent states you're in, baseline, planning, or moving, and then given that state, decodes the intended target. So I developed a hidden Markov model of this neural activity. So where the latent state now represents which of these 3 possible regimes of activity that we're in. So let me just show you what it looks like. So the model says, let's take neural activity in a 10 millisecond window and associate it with a state in our model. And here's actually a different representation of the model. This actually shows the details. We'll learn what our firing rate for this state is, and then we'll assume a Markov transition between states. So for this particular example, we might say that this activity was generated by states transitioning between baseline. The target comes on. Now we are in a planning state, and we have some generative model for this neural activity. And then the go cue comes on, and we transition into a pattern of activity that corresponds to movement. OK, so some of you may, those of you who are familiar with hidden Markov models may find this a little bit more explanatory. So we actually use the hidden states, in this case, not only to represent the regime of activity, but actually also the target that the animal or the user wishes to move to. So there are B baseline states, so corresponding to this activity that you have just in the background when you're not trying to move. And the results that I'm going to show you, B is 5. And then for each target that the user might wish to move to, we have one state that corresponds to planning activity, one state that corresponds to movement activity. And then for each state, we assume a Poisson process that generates spikes in a 10 millisecond window. So the parameters are the probabilities of state transition, which are sparse in the following way. Baseline states can transition through themselves, but once you transition to a planning state, you can only transition to the corresponding movement state. So these sorts of transitions are excluded. And here's the parameters. And we can learn this model using the traditional methods for training hidden Markov models. And then for decoding, once we have our model, and now we have some novel trace of neural activity, we can iteratively estimate this a posteriori probability. So the probability of being in state J at time t given all of the neural activity up to time t. And it turns out that this calculation, this a posteriori probability, is really nice and easy to calculate. That's why we like hidden Markov models, because this sort of state estimation is really easy with them. And it's actually really easy to implement as well. And then we'll pick which state we believe that we're in at any time to decode using maximum likelihood. So I just want to make that clear. So for those of you who are familiar with hidden Markov models, there's also a way to generate the most likely set of states. They use the Viterbi algorithm. So maybe some of you in communications are familiar with that. That's not what we're doing. We're actually propagating the state likelihoods for all of the states and then choosing the maximum one at any time. So this is what it looks like. So here's our example neural activity. And now here's the likelihood of each state in the model. So this block of gray here, hopefully, probably many of you can't see that. You'll just have to take my word for it, corresponds to the baseline states. And then this sort of checkerboard pattern is the planned state and the movement state corresponding to each of the 8 possible targets. So we can see that this is now the decoded likelihood that we see given this neural activity. So during this period, prior to the target onset and then a little bit afterwards, our model thinks that we're in a pattern of activity that corresponds to baseline. And then a few 100 milliseconds, probably about 100 milliseconds after target onset, we see a transition in the state likelihood. There's a little bit of uncertainty initially, and then we lock in, so we're almost 100% that we're in the state corresponding to planning to target 5 in this case, 6. And then about 100 milliseconds following the go cue, we transition into this moving, the state corresponding to moving to that target. OK, so what I just described still uses this information, this extraneous experimental information. I told you that what we wanted was an automated system for extracting which regime we're in. Well, we can do that really easily. So we can calculate the probability of being in any one of these regimes by just summing over the likelihood of being in the corresponding state. So for example, for the probability of currently being in a moving state, we can just sum over the state probabilities for all 8 of the possible moving states. This is really simple. And what's remarkable is that if we just set a threshold and then decode at this point, we have a system that works with very high accuracy. And let me show you the results. So let's say that we will just use planning activity. So we're going to ignore activity that the user generates corresponding to imagining moving to a particular target. We're just going to take that brief window of activity that corresponds to planning. That's what this blue curve represents. Now, what is this blue curve? It turns out that, as I said, as electrodes are implanted, this process of gliosis causes their quality of the recording to degrade. And eventually, we're no longer able to get good signal from many neurons. And so the number of neurons that we have available to our decoder decreases. So one way of evaluating how good an algorithm works is by looking at its robustness to this sort of degradation. So here, for example, is the robustness to degradation. But this is the decoding using all of the information. OK, now if we use also the activity up to the time where we detect that the user is now in a movement state, then we can see that our performance increases, and it's over 90%. It's close to 100%. Now, this was the first time anyone had ever done this, had designed a system which would automatically detect the state that the user was in addition to figuring out where it was that they wanted to move to. And if you think about it, it's actually a really important problem. If you have a neural interface that's on all the time, then all that sort of background activity that you're generating in your brain constantly sort of ends up corresponding to these sort of like drifting movements in your decoded arm position. And you don't really want your arm always moving. Sometimes you just want it still, right? And it's nice to be able to detect that these are cases where the arm is not intended to move, and then these are cases when the arm is supposed to move. But how does this compare to that original, so the cheating method where we take into account the known windows? Well, it turns out that obviously the performance of that is a little bit better, but actually here at the boundaries with all the neural information, it's not that much. OK, so in summary, in order to solve this problem, we had to identify some hidden states, that there was this baseline activity, this planning activity, and this movement activity. And then based on those hidden states, we were able to decode the target, and we were able to actually result in an interface that was controlled, and that actually used both this plan and this movement activity. And here's the corresponding paper. OK, so as I said, we actually also did a lot of work on decoding the trajectory of an intended movement. And it turns out that if you have a movement that's directed towards a goal, and you have information about that goal independent of the information about the thing that you're sort of tracking, that can improve your estimate of the trajectory. Yes?
Sorry. As you extend this work, is it possible to actually overlap different movements in planning, or planning and movement of different sequence of actions? So while you're actually moving to one place, you possibly are planning the next phase.
Yeah, I think that the answer to that is that you'd have to look at that neural activity and see if it's distinguishable. And my guess is that it is. And in either case, that would require that you are automatically able to segment this activity. So here's some papers about that. You guys are welcome to ask me about them later. So let me review. So I've told you about acquiring neural signals. I've told you about an algorithm that we might use if we were interested in decoding them. So now I want to give you a 4-slide introduction to the hippocampus, which is actually the brain area that I'm most interested in. And I've called this the memory center of the brain. So how do we remember? There is this case of this famous patient whose initials were HM. His name was actually Henry Molaison, but he only died a couple of years ago. So his name was anonymous for a long time. In 1957, so 50 years before he died, he had incurable epilepsy, so they removed both of his hippocampuses. So when he woke up, the neurosurgeons were surprised to find out that while his epilepsy had been cured, he had lost the ability to form new memories. So he clearly remembered everything that had happened up to a few weeks or a few months before his surgery, but then everything after that, he wasn't able to make new long-term memories. So you could walk into a room, have a conversation with him, and then walk out, and then come back in 15 minutes later, and he wouldn't remember having talked to you. So what we learned from him and from many similar observations since then, and a lot of experiments, is that the hippocampus, which is shown here in red in a human brain, and here in a rat brain, you can see that for rats, their hippocampus is much proportionally bigger than ours is. The hippocampus is critical for the consolidation of short-term memories. What do I mean by consolidation? I mean taking something that happens on a short time scale and saving it over a long period of time. And it's critical for two kinds of memory. Memory that connects things across representations and connects things through time. OK, that is not supposed to make sense, so I'm going to give you an example. OK, the best chocolate chip cookie that I ever ate came from the Arizmendi Bakery in San Francisco. Arizmendi chocolate chip cookies they're big, they're about the size of your hand. They have two kinds of chocolate, like this really rich dark chocolate and this milk chocolate. They're crunchy on the outside, soft on the inside. They cost $2, and Arizmendi was a Spanish priest who started a bunch of socialist cooperatives in the 1920s. So what I just told you was information about Arizmendi chocolate chip cookies that come from a whole variety of different sensory modalities, from different representations, from sort of different cortical, or sort of from different ways of thinking about a cookie. And it turns out to form that association, you need your hippocampus. So the hippocampus helps cue the long-term storage of this distributed memory, many different types of modalities of memory together as a function of time. But in addition to that, I remember, as I told you, the best chocolate chip cookie that I ever ate. And it wasn't, I'm not just saying that the best chocolate chip cookies come from Arizmendi. What I'm saying is that the best chocolate chip cookie that I ever ate, I remember the day that I ate it. I walked out of the store with this cookie and this cup of coffee. It was a very sunny day, which is unusual in San Francisco. As I was walking down the street, drinking the coffee, I remember thinking, wow, this is a really good cookie. So I broke off a piece of it and I ate it as I was walking back to work. And then I gobbled the whole thing up. And as I finished it, I said, wow, this was the best cookie I ever ate. So that I just described to you is a memory of a specific event that happened. And it links together a whole sequence of things that happened together in time and across sort of many different senses as well. So these two kinds of memory are both supported by the hippocampus. This association that sort of forms links between different modalities, and then also the memory of sequences, or a particular order of events that go into the formation of a memory of one particular story. Okay, so what do we see when we look at neurons in the hippocampus in rats? Do we see something that sort of corresponds to these different patterns of memory? Well, what we see is we see that about half of the neurons that we record in the main output of the hippocampus produce these things called place fields. What is a place field? Well, let's say a rat is moving in a linear environment like the ones that we use in our lab for experiments. So he's exploring this environment, and he gets to this point represented by this green oval. In this space on the track, and only in this space on the track, this cell represented by these green spikes fires with a very high rate, so it produces spikes very rapidly. But everywhere else, it's pretty quiet. And then let's say that he moves on to this space represented by this blue oval. Now a different cell fires in the same way, and then similarly in this purple oval. So we can see as he moves through the environment, the cells will spike in some order. So what are these place fields? What do they mean? Well, at some level, okay, they form a map. So if the rest of his brain is listening to his hippocampus, then his hippocampus is saying, I'm here now, okay, now I'm here, okay, now I'm here. But if you think about it, the definition of here is really an integration of a lot of sensory information, right? The fact that I'm standing here and receiving this pattern of light and this sort of acoustical properties is very different from the properties that I have when I'm here. And so at some level, it seems like that these place fields correspond to this associational cues that then would help the cortex learn to put all the things that represent here together. So this isn't the only pattern that we see in the hippocampus, however. We also see these high-frequency oscillations in the local field potential, mostly when the rat is sitting still and sometimes when he's moving slowly. And during these periods, which now the time scale is very important, they're much more rapid, we see a different pattern of firing. And let's sort of put the two together. So what's cool about these ripples is that during this period, the cells which fired in order as the rat ran through this environment fire in the same order, but on a super compressed time scale. So here we see these cells, same order. And we call this a replay event. It's as if the rat was running through, after he had ran through this environment, after he had experienced all this sensory stuff, he's sitting still now and he's thinking back, what have I just experienced? And he's putting it all together on a very compressed and rapid time scale. Okay, so that might be useful for navigation. You might be like, okay, well, how did I get here? But I think that this is also the key to understanding the memory of sequences, how we link all of our experience together. You could think of if each of these places represents an object with different properties that we've linked together across all these modalities, then a sequence of places would represent that sequence of things that went into my memory of the best cookie ever. And as I think about that memory, as I play it back, that sequence of events can get stored into my cortex for a long-term memory. So this is the question that has sort of intrigued me recently. Could these ripples be really something fundamental? Just like we had a pattern of neural activity that corresponded to an arm movement, could these ripples actually be the corresponding trajectory of memory? And so this is sort of the question that I'm sort of moving to trying to answer over the next few years. Can we prove this? Can we prove that these patterns of ripple replay are the elements of memory. And so finally, I want to talk to you about optogenetics and putting together this whole memory interface into one step. Okay, what are optogenetics? Well, the easiest example is channel rhodopsin. This is something that's new in neuroscience, by the way. So this is only, I think this was first discovered about 10 years ago, first started being used in neuroscience about 5 years ago. Okay, well, let's start with this word, optogenetics. The opto part refers to the fact that this ion channel rhodopsin 2, is sensitive to light. Turns out this comes from an algae, but if by some means we could get a neuron to express this ion channel in its membrane, well, if you remember, the way that neurons signal is by pulses across their membrane, so voltage pulses. So if we could express this ion channel and then expose that neuron to light, then we can make it signal. And so now what's weird is, okay, there's neurons in your eyes that are sensitive to light, but the rest of your brain isn't really. So what this means is that we could make neurons in some weird part of your brain spike by exposing them to blue light. And we use this general term instead of just talking about channel rhodopsin because in addition to this ion channel, there's other ones that inhibit neural activity. So if you expose those neurons to yellow light, then you can stop them from ever being active. And then there's others where you can use these tools to mimic the action of serotonin or dopamine or other neurotransmitters. So we're really excited about using these tools to sort of demonstrate that we can create particular patterns of activity. Now, the genetics part of the optogenetics comes from the fact that we use either like a genetic model like a mouse or a virus to actually make neurons express this ion channel, right? This normally comes from an algae. So we take the genetic sequence, we encode it into a virus, and we use that virus to infect particular neurons. And we can target the virus to particular kinds of cells or particular locations within the brain. And so we can actually get some really limited expression of this stimulation using genetic tools. Okay, so let me give you yes, so first let me show you that this works. So this is an animal. He's running around in an environment. This is a rat, and we've expressed channel rhodopsin in some neurons in his brain. And here are the spikes from one of these neurons. And you can see here this is being excited by a blue laser. The laser comes on, the neuron spikes with this really high fidelity, and then there fires a burst of activity, and then the light goes off, and the neuron goes quiet, and then it starts back up in its normal firing. And here you can see the waveform. So during this period, the neuron is producing spikes that are pretty much the same. Okay, so as I've described it, channel rhodopsin is good for exciting like large populations of neurons. And this is our third engineering challenge. So remember, our first engineering challenge was acquiring neural signals. Our second engineering challenge was decoding neural activity. Our third neural challenge is that we need some sort of really good photonics and electrode development to be able to put the light right next to the, sorry, I should have a picture here. But so what we'd really like to do is we'd like to put the light very close to the neurons that we want to record from in order to limit our activation to just cells that we are especially targeting. And another benefit of that is then instead of using a laser and requiring a lot of optical energy, we could use something like a diode that we could actually mount on a rat's head and power with a battery. And also would require some better genetics in order to do this, but that's a solvable problem. Okay, so I think in the 5 more minutes of my talk, I want to give you a brief tidbit of the science that we've done using this channel rhodopsin. So here's the hippocampus. The hippocampus is actually a little bit of a distributed circuit. There's 3 areas. Here's all the input coming in from all the sensory areas, and then it's going back out here. The output is area CA1. Then there's these 2 other areas, the dentate gyrus and area CA3. So we asked the question, is there a mechanism in this circuit? Is there something that's changing that allows it to support both that place cell activity and that ripple activity sort of simultaneously? The way that we did this was that we expressed this channel rhodopsin protein in the dentate gyrus. So this is, here's the cells of the dentate gyrus. Here's their projections to area CA3. There's this feed-forward projection. And then, of course, this projects on to area CA1. And here's just details of the molecular biology. And then we built this microdrive. So remember, microdrives with tetrodes. So here's our tetrodes. And then here's an optical fiber that aims the light next to the tetrodes in the hippocampus of the rat's head. And we implant this on his head, and then when we're recording, we plug in an optical fiber that goes to a laser and some amplifiers to go to our recording apparatus. Okay, so here's our question. And the way that we're going to answer this is by pulsing into the input of the dentate gyrus and recording here at the output. So at some level, what we're going to do is we're going to measure rapid changes in the impulse response of this circuit. So here's an impulse, and we're going to measure its response. All right, I'm not going to give you the details of how we get to this result, but here's the result. We found that behavior, in a very simple measure, just how fast the rat was moving, modulates the strength of these connections, and specifically these connections between area CA3 and CA1, and the ones that go recurrently into area CA3. And so what we find is that in slower and at times when the rat is moving slower, the strength of this connection is the strongest. So that means that the information that's going through the hippocampus, as opposed to information that's going through this little direct pathway directly to the output of the hippocampus, is favored. And in a novel environment, this effect is strongest, and then as something becomes less important to remember, we see less of an effect of behavior on the circuit. And then through a different means, we're able to actually measure the strength of this connection, and we find that as a function of behavior, we get this opposite effect. And it turns out that we think that place cell activity, which corresponds to these rapid associations between different modalities, probably depends most strongly on this projection. So what that means is that as a rat is running really quickly, he's accumulating a lot of evidence about what he's seeing. He's accumulating a lot of information about what he's seeing, and he needs to rapidly be able to detect that there's just different objects and make associations between those different objects. And then as he slows down, especially in a new place, he needs to reflect on what he's just seen, reflect on the experience that he's had, and so it favors this pattern of sequence learning. Okay, so this is now our memory interface. We need online spike decoding, we need some detection, and we need some channel rhodopsin to close the loop. So here's our hypothesis. We want to prove whether or not ripple replay equals memories. So our ripple is this atomic unit, this trajectory of memory. Okay, so there's two very easy tests, one of which we're doing and one of which that I'd like to do. So one is that we can just block all of these from ever occurring. If that was true, then you should be able to block the formation of all memories. And we've done, I'm going to show you, well, I'm not going to actually show you the results. The results from this are that this seems to work as expected, but because it's such a big hammer, it's really hard to know whether it's working for the reason that we think it is or for some other reason. Now, what I'd like to do is to detect particular patterns. So say that the rat went through this particular behavior. It'd be really nice to be able to say, okay, well, let's say if he was in place A and then place B, we're going to block his memory of place C by just stimulating at this point and causing a different neuron to fire. Then that would mean that his memory of that particular behavior would be modified. So that's going to require us to be able to do fast decoding and detection. So this is the very simple system that we built right now. It just detects these oscillations and it stimulates. This is just showing that we can do this online and in real time. Well, not real time, but with a latency that is acceptable but could be a lot shorter. So here's the challenges to making this work. We need to improve our detection of this system. We would like, as I said, better light delivery. We would like to use some of the newer optogenetic techniques, and we would like to do this all mounted on the head so that we could actually erase memories even as the rat went back into his home and sat there and thought about what he had done earlier that day. We want to be able to manipulate the memories the whole time. So here's a summary of what I just told you. And finally, here's the principle. I think that we can understand the brain by interacting with it. Now, that's a principle for neuroscience. As I said, this is my neuroscience theme for this talk. And I hope that you believe me that in solving this question, in proving that we can actually detect and decode individual memories, that will lead to progress in computer science and engineering. I've shown you some challenges in signal processing, and I've suggested that there are new challenges for low power, high computation, embedded computing. And I think that we can actually learn from the hippocampus perhaps how to do things that we would like computers to do, but that's a topic for another talk, so feel free to ask me about it at the end. And so finally, I just want to thank Teresa and Krishna, who are great advisors as a graduate student, and my current PI, Lauren. And then I've done some work, some of the the data that I presented is work that I've done with other with a grad student in his lab and another postdoc. And my funding, the Helen Hay Whitney Foundation, the NIH Sloan Swartz Foundation, and Marco C2S2 when I was a grad student. All right, and I'd be happy to take questions now. And I just want to apologize because that was like, there's a lot of detail. And hopefully if you just got a little tidbit, the brain is exciting, maybe we could decode memory, that's enough to take away, and you can ask me questions later. Yes?
So it reminds me of the Eternal Sunshine of the Spotless Mind, that
Yeah.
movie. And what are your thoughts on those sort of applications of this research?
Erasing people's memories?
Yeah.
You know, it's actually, like, you laugh, but that's a really profound question. Let me tell you why. PTSD. So for some people, memory of traumatic events can be debilitating. And there's actually some evidence that memories of events, like the kind that I described you, may never actually be fully formed in a long-term sense. They may constantly require the hippocampus through your whole lifetime. So this guy, HM, who had the hippocampal lesions, there's some evidence that he had trouble with his memory of sort of these sequences of events. And there's a lot of evidence that you sort of, when you remember something, that you restore it via your hippocampus in your cortex. And so it may be that you can actually help somebody with a PTSD type disorder by disrupting the reconsolidation of that memory. And it might actually be a very useful thing. I think that there may be psychiatric ways that you could do that, potentially with drugs, as opposed to using the optogenetics in a person. But it may be that for somebody for whom it was really debilitating, that would actually be really useful. So, yeah, good question.
So Caleb will be down here for another 20 minutes. I encourage you to come down and chat with him. Thank you for coming, and let's thank Caleb again.